welcome you all to presentation, which is pretty exciting. I am very excited to hear actually Lucina's presentation. She is right now working at University of Miami. You probably already Googled her to know all the details about her and what she does, etc., etc. Okay, so if I try to pronounce her name correctly, it will be Lucina Uddin, right? That's probably the correct pronunciation, right? In my own language, actually, in Bengali.
Exactly
Okay.
correct. Exactly correct.
Okay. Okay. And Lucina actually has a fantastic career. She has contributed immensely in our understanding of cognitive neuroscience since early career on. She holds a PhD in psychology and cognitive neuroscience from UCLA. Following that, like she did her 2 postdoctoral tenures, one in New York University in the Child Institute, and followed by that in the Stanford School of Medicine, where she was also a teaching instructor. She has published actually in number of high impact journals in very influential articles specifically for kind of expanding our understanding of the salience networks of the human brain and providing a salience network model of the human brain and testing these with variety of neuroimaging experiments and empirical evidences to substantiate that hypothesis. She is also currently the editorial board member of number of reputed journals, including namely Biological Psychiatry, Neuroimage, Network Neuroscience, etc., etc. Among her notable awards, she is one of the recipient of the Young Investigators Award 2017 from Organization of Human Brain Mapping, and one of the lesser known facts that she was the recipient among previous 21 years, I think, the first recipient who is not a white male. Is that correct? Yeah. And he has also received an award from International Society for Autism for Innovation in Autism Research in 2013. And also her studies is more closely works on understanding the large-scale brain networks and their connectivity and dynamics. She currently directs a lab in University of Miami, which is also called like Brain Connectivity and Cognition Lab. And what she often says, actually, I found out that she likes to go to international conferences, meet with like international collaborators, and come back with a renewed enthusiasm in order to do intense and hard research and solve the hard science problems. Okay, so I feel that after this talk, we'll also get sort of like, renewed enthusiasm in order to tackle these problems in our own lab. So with that, I would ask Lucina to take the podium. Thank you, Lucina.
Thank you very much, Dipanjan. It's my pleasure to be here today virtually. I wish I could be there in person. And thank you so much for this wonderful introduction. I don't have to take too much time to tell you who am I, because you already heard it from this introduction. But just to give you a little bit of background for, I appreciate that everyone is joining at such a late hour, which for me, of course, is early morning in California. But I was actually born in Chittagong in Bangladesh when, my parents are from there, and I was born there as well. So we came to the United States when I was 6 months old. So actually, I was very young when I first came. And I spent, many years as an undergraduate and graduate student at UCLA working in neuroscience and psychology, and then went on to work for a few years in New York University, where I first started to get interested in resting state fMRI and functional connectivity in human connectomics research. And then finally went to Stanford University to do some more postdoctoral work with Vinod Menon, and stayed there for a number of years, as well as faculty. And what was interesting is during that time in 2010, I had an opportunity to go back actually to Chittagong for to teach a course for 1 semester. So I went to the Asian University for Women and taught a psychology course there. You can see my students. And that was really a nice experience for me because after spending most of my life in the US, I had a chance to go back really to my home city and spend some time there. And then, so during, after I came back, I actually got my first position at the University of Miami in 2014, where I started the Brain Connectivity and Cognition Lab, and I'm still there now 6 years later, with one exception, except that now I'm in San Diego because this is my sabbatical year. So I'm spending some time here again, back in Southern California. And because of this COVID situation, as you all know, we have plenty of opportunities for virtual meetings, if not any in-person meetings, unfortunately. So I'm very happy to be here today to have a chance to share some of our work. Most of what I'll tell you today is focused on the concept of flexibility. We know in particular in children with autism that have difficulty behaving flexibly in day-to-day life situations. Maybe they prefer to wear one pair of socks or one type of color, or to eat one type of food. Often you hear about this insistence on sameness and need for habit and routine. And this inflexibility is part of the disorder, and also you see it in other conditions across the lifespan. But nowadays, I can tell you, I don't have to tell you how important flexibility is, because we all noticed that due to the pandemic, everything in our lives has become flexible and uncertain. And from day to day, we might have to change our habits or change our behaviors because the situation is uncertain and we don't know when we'll be going back to work in a normal fashion. But flexibility is very important for, for success academically and professionally. And so we're very interested then in the brain systems that enable flexible behavior and cognition. And what we do in the lab is we study brain network development broadly and how that relates to cognitive development in areas mostly of executive function, planning for the future, and control of behavior. We're also interested in how the disruptions to particular large-scale network interactions can be related to atypical cognitive development as well. So we're interested in children, adolescents, and adults, really lifespan studies of brain network organization. And we tend to work in 3 domains at once, really looking at neurotypical adult brains using neuroimaging, as well as typically developing brains, and also clinical populations, including autism. And the approach we take is to use multimodal neuroimaging, both functional and structural, diffusion-weighted imaging, and connectivity modeling approaches. I think many of you are really at the forefront in terms of the methods development, so I think you don't have to hear it from me, you already know these things. And the foundations theoretically come from the emerging field of network neuroscience. And even though we think often of functional MRI as the major tool, even using neuropsychological approaches, you can see from lesion studies that cognition is subserved by interconnected neural networks. So any complex behavior is mapped at the level of multifocal neural systems, not just a specific anatomical site referring to one function. This is even seen back in the early 90s, before mainstream neuroimaging approaches were commonly used. And there's also this nice idea of neural context from Randy McIntosh, that the functional relevance of a brain area depends on the status of other connected areas. So if you see the nodes in gray in figure A, you can see they might behave differently in the context when these black nodes are coactive, versus in B, the gray nodes have a different set of coactivation, and that context may affect their function. And finally, we're very interested, as many are these days, in network dynamics and changes over time. The idea that a brain network needs to be understood in terms of the interactions between multiple brain regions as they unfold temporally. So as we'll all see that there's many things about the timing of events that we're now starting to understand as being important for flexibility more broadly. And ever since Bharat Biswal in 1995 published his paper showing coherent fluctuations in the motor cortex, people have really taken advantage of resting state functional MRI to map the organization and integrity of large-scale brain networks, which you can do when participants are just resting in the scanner, and you still see these coherent fluctuations at a very slow scale. And you can see systems like that are similar to those you would see during a cognitive task. So maybe a language system, a memory network, attention networks. All of these things seem to be, there's a signature in the brain that spontaneously, maintains these kinds of networks even when when we're offline, even when we're not using them. So of course, this has changed the field a lot and changed network neuroscience dramatically in the last 10 years. And in the laboratory, I come from a psychology background and neuroscience background where people come up with very complicated tasks in order to test some kind of function. So in adults, there's a lot of task switching kind of paradigms where there's a lot of high working memory load and you're asked to perform one task when there's one condition and a different task when there's a different condition. But when you try to test children, as they behave flexibly, they don't have as high of working memory capacity. So you often have to come up with simpler ways to test flexibility that don't involve so much holding information, working memory. And we know there's many other constructs or processes that you can't really isolate cognitive flexibility per se, because it's involving processes like attention, inhibition, switching, and working memory, which engage a lot of the same lateral frontoparietal systems. Broadly. So it's very hard to tease apart which brain system or network or set of regions is important for flexibility per se, although there's some evidence that the inferior frontal junction in particular seems to be very much involved in flexibility, task switching kinds of paradigms. But there's many systems in these frontoparietal and singular and thriller networks that are involved in flexible behaviors. So it's not a very simple story. We tried to come up with one task to do in the scanner, which really came very close to picking out this flexibility aspect from all the other aspects, attention and working memory. And what we did here was just ask participants to pick two things that go together. This is a flexible item selection task. And you might pick these, the boat and the rabbit because they're both blue. Then you ask them, pick two other things that go together, and they might pick the two bottom panels because they're both rabbits, and then say, pick another two things that go together, and you might pick the first and the fourth panel because they both show one object. So objects can match on dimension and color, or several dimensions, color, shape, size, and you have to sort of flexibly pay attention to one attribute versus another in order to do this task successfully. And it's been done in children and adults and psychometrically validated. And we know sort of the age at which you become more proficient at this task. So there's a lot of psychology from developmental psychology background on this task. But when you put it in the scanner, of course, you have to come up with a condition that you can subtract to find out what brain regions are more responsive to flexibility. So we just have the participants press the buttons corresponding to the box that are outlined here. So they're not doing a task, they're just pressing the buttons and following along, essentially. So when you do this flexibility minus control in the scanner, you can do it in this manner that I'm showing here on the left. Participants have to pick 3 selections. And it turns out that we had to validate this against their performance outside the scanner, and there's a high correlation between their computer task performance and their fMRI task performance. And the participants really become more efficient over time. So if you ask them to do 4 runs of this task, if you calculate a metric that combines accuracy and reaction time, they do get better over time, as you can see towards the end in run 4 here. Now we know that they're going to activate these frontoparietal systems, basal ganglia, singular insular systems, and cerebellum. That should be not surprising. Here you see very high levels of activation if you subtract flexibility from control trials. And this is a group of 30 adult subjects. This is work from my former graduate student, Ajani. What is more interesting is that there's levels of task modulated connectivity and great individual variability in the networks that are used to do this task. So when you see the group activation levels, it just says, okay, all these areas are involved. But here we're using a group iterative multiple model estimation, which uses a structural equation modeling framework to look at directed task modulated functional connectivity between regions of interest. So here, these are the regions that were most active in the task and the sort of relationships among them at the group level and at individual levels. There's a lot of variability in how this network actually completes the task. That's one point. And the other point is the the sort of main effect was found in the inferior frontal gyrus, which seems to be sort of initially activated during flexibility trials and seems to be sort of causing the activation downstream in other regions of the network that we saw here. So there's ways of getting into the individual contributions of these different brain regions to this kind of flexibility task. Now, the reason we developed this was because we want to then find some way to test children with autism and look at typical development of this process. So we have another sort of branch of work in the lab that looks at the development of flexibility, both cognitive flexibility and brain network flexibility. We know that developmental trajectories follow a protracted period. So attentional control, flexibility, and goal setting all show this protracted. Excuse me one sec. Okay, so we see these developmental trajectories show protracted development. And we also have done some network analyses comparing children and adults to find, for example, things like the insular output to dorsolateral prefrontal cortex and anterior cingulate changes with age. So it's a stronger effective connectivity in adults compared with children, which is accompanied by stronger structural connectivity between some of these brain regions. And we also do sort of whole brain analyses to look at how particular networks, here's the salience network, spatial topography as it changes over the lifespan, and network interactions as they change from, this is, using age as a continuous variable and looking at strengthening and weakening of between network connections over lifespan. And also look at things like brain signal variability and how that changes from age 6 to 85. Some regions become more variable in their bold signal, some become less variable over the lifespan. So this is another branch of work in our lab where we just look very much at how brain networks develop and how that underlies some of these cognitive differences across the lifespan. So here in children, back to the flexible item selection test, children also activate frontoparietal systems similar to what you saw in adults. This was a registered report we started a few years ago. What happens is that we started this study and then we couldn't get as much far into the data collection as we wanted to because in March our scanner was no longer usable because of the pandemic. So we have collected data from about 30 typically developing children between the age of 9 and 12. And we only collected, I think it was 16 or 18 children with autism completing this task. So we haven't finished this data analysis yet. And unfortunately, just like the rest of the world, we had to stop our data collection on this just in the middle of in the middle of finishing the study. So nevertheless, we'll try to analyze the data that we have and to complete this registered report. But what I wanted to show you basically is that we think that cognitive flexibility involves coordination among the brain regions that are important for processes in executive functions, including inhibition and working memory, in combination with attention, salience detection systems. And adults engage the lateral frontoparietal brain regions as well as a salience or mid-singular insular network during the flexible item selection. And we saw in the adults that task modulated connectivity of the inferior frontal junction in particular seems to be associated with the flexible behaviors here. So hopefully we'll be able to show you the results of this pre-registered study comparing children with autism and typically developing children. But just like many others, we were disappointed that we couldn't finish this, this project and collect all the data that we wanted to, unfortunately. So more broadly, we're interested in the brain dynamics that relate to flexible behaviors. And you can do a lot of this work just using the Human Connectome Project data, which is available now. And one way to look at changes in functional connectivity on the second to second or moment to moment is to, instead of looking at whole brain functional connectomes, you can break the time periods up into sliding windows, for example, 45-second windows, and create whole brain connectivity matrices at each of these windows, and then do some type of clustering in order to find out what the dominant patterns are. And you can say, well, there's you perhaps label them as different brain states or functional connectivity states. And the nice thing about that kind of analysis is then you can quantify things like the frequency of occurrence of a particular functional connectivity pattern. You can quantify the dwell time. How long does this pattern in the brain stick around or stay in the brain? And how many transitions are there between brain states? This is a figure adapted from Vince Calhoun's group, and we've been using their GIF toolbox to do this type of analysis applied to restings data from RAI data from the Human Connectome project. So Jason Nomi in the lab has spent a lot of time using ICA to do whole brain parcellation. And when he did this study, he took about 200 subjects with 1 hour of restings data from RAI data from Human Connectome. And the nice thing also in that data set is you have so many different cognitive processes that have been assessed outside of the scanner. So you have a lot of restings data from RAI data and TASCA from RAI data. In addition to that, you have outside of the scanner processing speed, inhibition performance, cognitive flexibility performance, and Wisconsin card sort, fluid intelligence, working memory. It turns out that if you take the restings data from RAI data and look at the dominant states using this dynamic functional connectivity sliding window approach, there's some states that happen more frequently, about 36% of the time on the left, and other states that happen very infrequently, about 10% of the time on the right. And they're sort of more strongly correlated states occur less frequently, whereas the weak connectivity states occur more frequently. And it turns out that those individuals who perform better on working memory spend sort of more time in the state 1 and less in state 5. And something similar happens with cognitive flexibility. The performance is correlated with how much time is spent in state 2 versus time spent in state 3. So this suggests that there's something about the whole brain connectivity pattern that predisposes individuals to be more or less adept at executive function. So greater cognitive flexibility is then associated with the propensity to occupy the more frequently occurring brain configurations that we saw were characterized by attenuated correlation and greater functional connectivity variability. And this is just one example of how you could use a dynamic functional connectivity approach to reveal relationships between brain dynamics and flexible cognition in the typical adults. So that was a whole brain study, but as you heard from the introduction, we've been interested in many ways of parsing what are the main neurocognitive networks in the brain that you see, in a sense, are ubiquitous, which means they are always there, and you see them across very different kinds of paradigms. We've pushed this model for a while that the salience network anchored in the anterior insula and anterior cingulate cortex seems to be involved in dynamic switching between the default mode network with nodes in the posterior cingulate cortex and medial prefrontal cortex, and switching to the central executive network with key nodes in posterior parietal and in the dorsolateral prefrontal cortex. And we think that because we see the salience network tends to come online first in terms of its activation, and if you use Granger causal modeling, for example, you can predict what will happen in default mode and central executive based on the time series information in the salience network. So this has been something we've looked at for some time now. And so there's something about these three networks that you can see them in some form across a lot of different cognitive processes. And the breakdown in disorders across the lifespan is evident that there's some transdiagnostic information perhaps in salience network integrity that can be used to, it has been adapted a lot in the psychiatry literature as a biomarker potentially. So what we did, we had a student from Germany come visit our lab for a year, and she had some nice data from about 100 participants as they performed an attention task, which would involve self-control. So it's really a simple paradigm where the person is cued and they're supposed to say which letter comes on the screen. It's either a letter E or letter F. And there's also eye tracking going on. And so you can see the participant is moving their eyes to the target. But at the same time, there's distractors, sometimes very interesting distractors that make the eyes go in the opposite direction. And so oftentimes the participant misses the task altogether, which is to say which letter pops up on the screen. So the eye tracking is very interesting to quantify the attention here. And you can find in a task like this that when there's a distractor there, the reaction time is slowed down and the error rates go up, and the mean gaze distance, how far the eyes travel from the target, is very much affected by these distractors, as you can imagine. So what's interesting is that if you look at the salience network dynamics just in the resting state, outside of this task performance, there's some very different ways in which the salience network seems to interact with the rest of the brain. This is Rosa here on the left who did this study. And what actually it becomes apparent is there's one state, salient state 4 here, where the salience network is actually negatively correlated with the visual network. You can see these blue squares here indicate low functional connectivity between salience and visual. And what's really interesting is that those individuals who spend less time in this state, or who show fewer occurrences of this state, are the ones who are better able to inhibit the distractor. They're better able to stay on task and ignore the distracting images. So again, this is just a way to relate salience network dynamics. And interestingly, it was the salience network, but not the frontoparietal control network, that was correlated with the capacity to resist these tempting distractor images. So this is just to show that you can use this kind of dynamic analysis to reveal individual differences in self-control capacity. Now, because we spend so much time on the salience network, it's important to understand a little bit about the anatomy and functional specificity of different parts of the insula. And in particular, if you look at the cognitive neuroscience literature, the insula is activated across tasks of emotion, empathy, pain, awareness, interoception, somatosensory processes, conflict, switching, attention, inhibition. Basically anything you can imagine will activate the insula. So the question is, why is this such a non-specific kind of reaction? And if you look at some of the functional parcellation literature, Ben Dean, Luke Chang have shown that you can use whole brain functional connectivity to parse the insula into at least 3 subdivisions. A lot of parcellations have now broken it even further, up to 20 or so subdivisions. But here's just an example of one tripartite subdivision consisting of a dorsal anterior insula, the ventral anterior insula, and a posterior insula subdivision. And if you look at what tasks most are associated with these different subdivisions, again, in Luke Chang's meta-analysis, he finds that the ventral anterior insula tends to show up in studies where these terms on the red terms are used. So anxiety, emotion, olfaction, sort of affect. We did a connectivity modeling across a large database. And again, you can see that when the ventral anterior insula is active, the limbic regions, sort of, you can see orbitofrontal here a bit. What activates, it tends to be the studies in higher level cognitive processing, error processing, inhibition, switching, etc. And if you look on the right, these frontoparietal regions tend to be co-active with the dorsal anterior insula, frontoparietal association regions. And if you look at what regions tend to co-vary with the posterior insula, it's really the studies involved in pain and somatosensory types of things. And on the right, the somatosensory cortices are what are co-activated with the insula, the posterior insula. So this suggests, some subdivisional structure and distinction of different subdivisions, but at the same time, all of these subdivisions are active across all of these different kinds of tasks. So there's a common functional fingerprint as well. So one thing we did with Jason Omi here again was look at whether these different insula subdivisions show different patterns of functional connectivity dynamics that might explain these different functional roles. It's impossible to tell from the figure what's going on here, but we looked at dynamic functional connectivity of the different subdivisions of the insula and found different functional connectivity states 1 through 5 again. And in some states, all the different insula subdivisions tend to be functionally correlated with all the rest of the brain in a similar pattern, like in state 3 in the middle. Whereas in other states, like state 2, that are very infrequently occurring, the insular subdivisions vary in their connectivity patterns with the rest of the brain. So they diverge from each other. And what's interesting about the dorsal anterior insula is that it's more different between states than the other subdivisions. So middle, posterior, ventral, all those other insular subdivisions tend to be more or less similar in different functional connectivity states, whereas the dorsal anterior insula shows the most variability in its connectivity patterns with the rest of the brain. And if you look again at the structural connections that are unique to the dorsal anterior insula using Human Connectome Project data, you can find some unique projections to frontal cortices and thalamus as well that you don't see for the other subdivisions. So this suggests that the insular cortex in the adult brain can be parcellated into several subdivisions, but at least here I'm talking about a dorsal, ventral, and posterior that seem to be involved in cognitive control, emotion, and sensation differentially to some extent. And what's interesting about the dorsal anterior insula is that it exhibits the most variable patterns of functional connections, as well as unique structural connections that we think might be what enables it to do the kind of flexibility, the functional flexibility that we see across many tasks. And here's another one of those projects that's been put on hold due to COVID. So it's very interesting if you can collect data from intracranial depth electrodes. These are usually implanted in patients who are being monitored for epileptic seizures in the hospital, but oftentimes you can conduct tasks on these patients as they're waiting in the hospital. You can collect very high resolution, high spatial and temporal resolution data because the electrodes are placed directly on the cortex. So a lot of network analyses have been enabled by these types of methods. Excuse me. So what we see on the left is just some work that's already been published by different groups doing network analyses from these types of very high resolution data. And I was excited to start a collaboration last year with the University of Western Ontario. They have a hospital where they implant depth electrodes in the insula. They're one of the only hospitals in North America that does this kind of monitoring. So they're specialists in the insula epilepsies. And what they often do is implant 3 different electrodes in sort of the anterior, mid, and posterior insula. So the goal of this collaboration was to be able to look at these data to find out if what we're seeing in the functional MRI studies, the differentiation holds up at this very fine scale, and to really be able to test some of these hypotheses about the timing of activation in different subregions and how they affect whole brain dynamics. Of course, like everything else, this is a project, again, that's on hold that we're hoping to start up again soon. So the sort of end goal in all of this is to reach some translational kinds of findings that can be of interest to those interested in neurodevelopmental disorders. And the reason I'm so interested in cognitive flexibility partly is because I think it's one of the aspects of autism that is less studied relative to the social and communication aspects of the disorder. And one thing you'll note is that there's some sort of poor outcome statistics that suggest that children with autism, they don't sort of live independently, or they always end up staying at home with a parent or guardian, even into adulthood for the most part. And this is in contrast to these other developmental conditions that have seen better outcomes in terms of independent living. If you look again at unemployment, that's also a very poor statistic. So about 80% of young adults with autism remain unemployed or underemployed. And I think part of these statistics have to do with not only the social difficulties that are experienced by individuals, but also these flexibility deficits and executive function deficits. So, just not being able to change your behavior in a work setting might, also lead to these kind of lower employment rates. There's a few targeted treatments and interventions that have been developed to improve flexibility and executive function more broadly. And those are now beginning to be implemented in classroom settings and with some success. What we're interested in is the fact is there's some heterogeneity here. So not every child with autism shows executive function deficits. In fact, if you do a latent profile analysis, and look at a mixed group of children. Here we had a group in collaboration with Kennedy Krieger Institute. We got data from children with autism, children with ADHD, attention deficit, as well as children with comorbid autism and ADHD. And if you do a latent profile analysis looking at the behavior rating inventory of executive function, which is a parent report measure, you actually find 3 classes of executive function profiles, above average, below average, and average. If you look at that average profile, not everyone who is in the average group is typically developing. In fact, there's a big proportion of children with autism in the average group, as well as ADHD. In fact, those who are the most impaired are those who have comorbid, both autism and ADHD, show the most impairments in executive function. So this was a latent profile analysis that we also replicated in another study of just autism and typical development. So just having the disorder of the diagnosis doesn't mean you'll necessarily have executive function deficits, but it does, if you look at those who are the most impaired, they tend to be those that are diagnosed. Oops, that was an undergraduate, Haryana, in our lab, who found this in a replicated sample. So this heterogeneity, we think, could be explained with underlying brain dynamics. So one hypothesis is that perhaps there's fewer transitions between brain states in children and adults with autism that correlate with this reduced behavioral flexibility. Again, Jason did this work looking at some of the data from the Autism Brain Imaging Data Exchange, a large data sharing initiative led by Adriano DiMartino at New York University. He found evidence in this sample for 4 different connectivity states, and initially some initial evidence that there are fewer transitions between brain states in the individuals with autism, just as we predicted. If you look again, that was using the sliding window analysis, but there's many ways of looking at brain dynamics. This particular one, called coactivation pattern analysis, instead of using windows, does a more time point by time point breakdown of whole brain coactivation patterns and assigns each point to a particular brain state. At the end of the day, you can get the same metrics, which is frequency of occurrence of a particular brain state, dwell time in a particular brain state, as well as state transition. So you can, again, get these interesting brain dynamic profiles without the use of sliding windows. So what one of our students, Emily, just published this paper this year, used the coactivation pattern analysis to look specifically at the salience network, which we're interested in for obvious reasons. And she found that there's different patterns of coactivation, with the salience and the rest of these brain networks. And there's one in particular, state 2, which is characterized by coactivation of salience with all the other networks here, central executive and default as well. This pattern shows a higher frequency of occurrence in typical individuals compared with their typical kids compared with the children with autism spectrum disorder. If you look at this salience network, salience network dynamics in the context of a flexibility task, here we just pick the nodes of the 3 networks that I've been talking about: salience, central executive, and default mode. You can also find that across the period of a task, children with autism, they have a harder time towards the end, like the last run of the task. And towards the end of the task, they actually need to engage more of the lateral frontoparietal networks just in order to even be able to perform at the same level as their typically developing peers. So this was I guess, 2 papers, one in autism research and one in Neuroimage Clinical this year, that look at both the activation and the dynamics of the brain during these types of pretty simple flexibility tasks, to show that children with autism have a different way of processing the same information in order to reach a particular behavioral performance level. So we think, in general, dynamic functional connectivity approaches have the potential to reveal how atypical patterns of brain dynamics, and particularly related to reduced flexibility or transitions between brain states, in these neurodevelopmental disorders like autism, which are really characterized by inflexible cognition. And the extent to which these brain dynamics are related to individual differences in flexible behaviors is the current work, current set of questions that our lab is working on. So we, have the ultimate goal of translating this into, individual subject prediction, but there's a long way to go before that. So we have a review here with Manish Sagar, who's at Stanford University, and he talks about deep phenotyping of individuals and how that will be necessary in order to really translate these kinds of neuroimaging metrics into the clinic. We think we're a long way off from this at present. But this is what the things like the National Institute of Mental Health have been speaking of, stratifying data using the research domain criteria. They've been talking about this for a number of years now, how we can, instead of going from the diagnostic and statistic manual purely based on symptom-based categories, that we can perhaps integrate data across genetics, brain activity, physiology, and other processes to get data-driven clusters of individuals who then might be better served by a particular treatment or a particular, clinical practice. The reason I bring this figure here is because I like to note how the paper from Inside brain these kinds of transdiagnostic types of approaches. And I don't know if they meant anything specific here by putting this in their figure, or if they really thought the insular cortex is important, but I think it's important for all the reasons that I've shared with you today. So it's something that I haven't talked with you about here, but if you look at insular cortex activation and connectivity, it tends to be distinguished in autism and anxiety and depression and schizophrenia and frontotemporal dementia. It's just not specific to any disorder, but there's a lot of information within that brain region that seems to index when there is some kind of pathology. So I just think it's a useful brain area to look at if you're going to look at any. And the thing we're doing now, which is a complete departure from anything we've done before. So Celia Romero in our lab noticed that a lot of our participants are Spanish English bilingual because we do our recruitment in South Florida in Miami. And Spanish is really the second most spoken language, commonly spoken language in the United States. And there's been some evidence from the executive function literature that those individuals who speak another language can show some cognitive advantages. So in particular, this is known as the bilingual executive function advantage. You don't usually see this in typically developing kids or in typical adults, but in some groups like those with low socioeconomic status or older adults, older aging, you see these protective effects of bilingualism, whereas those who speak more than one language tend to have preserved executive function skills. So there's something interesting to explore here about whether bilingualism is a natural intervention to improve flexibility more broadly. And there isn't, there's still a lot of controversy in the field about whether this is the case or not, but we're very interested in studying this in the context of autism. Clinicians used to say, well, you should only teach a child one language because it will confuse them to teach two at once. And we know that's just not true anymore. There's no evidence that there's any detriment to learning two languages. And of course, it's only in the US that this question even comes up, because all around the world, everybody speaks multiple languages. So you wouldn't even think to ask, is it good to speak more than one language? But in the US, there was often this idea that in communication disorders, you should only teach one language at a time. But in autism, there's now been evidence that bilingualism might actually mitigate set shifting difficulties. It seems there's no harm, at least in teaching two languages in the home. And there may even be these benefits in terms of increasing flexibility more broadly. So there's some of this work just started to come out that those kids who are dual language speakers might even show reduced executive function problems. So what we, just because we live where we live, we found that many of our individuals in our studies were bilingual. They spoke Spanish and English in the home. So Celia Romero looked at these parent reports of executive function and found that just as the literature suggests, in typically developing kids, there's no real difference between monolingual and bilingual kids with regards to their parent report executive function measures. But in kids with autism, in our small sample here, those who were bilingual did seem to show a little bit better executive function scores than those who were monolingual. This is, of course, very preliminary initial evidence, but we're excited to to be following this up. So in general, these are the studies that our lab has been conducting and continues to work on in terms of trying to understand the brain basis of flexible behaviors and the different factors that can contribute to enhancing flexibility across the lifespan. And we hope that this will lead us to the goal of precision medicine and trying to find out how we can focus on getting children to attend to their interactions with the world and less and to be more flexible in their daily activities in order to promote optimal outcomes. This is a book, in case anyone is interested to hear a little bit more about the Salience Network per se. And as we heard earlier, we do a lot of collaborative work with individuals all over the world, and I'm always happy to expand that type of collaboration. And this is the network of co-authors right now, but I hope it gets even more complicated as the years go by. This is the group of individuals at University of Miami who did all the work that I've been talking to you about today. This picture is old, obviously, because we no longer gather in person, but hopefully we will again soon. And these are some of the individuals that have worked on all the studies I shared with you, as well as the funding sources. And yeah, everything is always a collaborative effort, so we can never do this alone. And this is San Diego, where I'm currently staying for sabbatical, and I think I have finished in time so that we can have some time for questions. Thanks very much.